Thanks for standing by and welcome to the Chimeric Therapeutics Investor Webinar, discussing the positive initial phase 1 clinical data from its chlorotoxin CAR-T technology. All participants are in a listen-only mode. There will be an initial presentation for approximately 15 minutes, followed by Q&A. If you have a question you'd like to submit, please feel free to type it in using the Q&A function within Zoom. Presenting on behalf of Chimeric Therapeutics today, we have CEO Jennifer Chow, and also on the line is Executive Chairman Paul Hopper. To begin, I'll hand it over to Jennifer. Please go ahead.
Thanks, Matt. Good morning, everybody, and happy Monday. I am very pleased to be with you this morning to share with you the first data that we have seen come through for our chlorotoxin CAR-T. So just, I think about an hour ago, there was a release that highlighted 3 key elements of the data that was released from embargo earlier this weekend. Those 3 sets of data are highlighted here. The first is that although we were not expecting or looking for efficacy at this low dose rate, we're very pleased to say that we saw a 75% disease control rate in the 4 patients that were treated. And so 3 out of the 4 patients. We saw no dose-limiting toxicities and that the CAR-T cells were generally well tolerated by all 4 patients. And importantly, we saw bioactivity. So we did see the CAR-T cells persist in the patients that they were given to. So before I go into each of these aspects in a little bit more detail to give you a little bit more context to them, I just want to take a quick step backwards to remind everybody about chlorotoxin and where we are with this trial and what this particular data set was looking at. So chlorotoxin, as many people know, was preclinically designed and studied for glioblastoma. And that is without a doubt one of the most lethal types of cancer. Patients with glioblastoma really only have that expected survival of about 12 months. The patient population that we are studying in our trial is actually a recurrent glioblastoma patient group. So they actually were diagnosed with glioblastoma. They were given an initial therapy that didn't work. They progressed. And now they're coming back for additional therapy. The survival of that particular patient group is less than 6 months. So really, this is a group that needs better therapeutics very desperately. Chlorotoxin, as many of you know, is derived from a deathstalker scorpion venom, and that makes it quite unique. And that is the element that binds to the glioblastoma cells in patients. And what we saw preclinically was that chlorotoxin binds exceptionally well. It binds better than things like IL-13 or HER2, and then it actually is able to kill those cells off better in our preclinical models as well. So that gave us a lot of encouragement as we headed into the clinic. We hold the exclusive license on this asset, and we have strong IP through to 2036. So the trial that is underway is a phase 1 dose escalation trial. The objective of this trial is really first and foremost to assess the safety of putting chlorotoxin CAR-T cells into patients, and then to determine what is the maximum dose a patient can tolerate safely, so we know what is the dose where we get the best efficacy with the least adverse events as we move into our phase 2. This particular trial is designed to recruit about 18 to 36 patients over 2 months. And it's in 4 different dose levels. And you see those 4 different dose levels highlighted here. Dose level 1 started giving patients 44 million cells through 1 single route of administration. We put a catheter in the patient's brain and the cells were administered directly into the tumor cavity through that catheter. That particular dose level recruited 4 patients, and those are the patients whose data is highlighted in the rest of the data set and in the data that was released on the weekend. The trial has actually progressed, and we're now in dose level 2, where we're now giving patients double that number of cells. We're giving them 88 million cells, and we're giving them those cells not only through that intratumoral cavity, but through a second catheter that's actually placed in their brain, so they can have intraventricular administration of cells as well. As we finalize this dose level, we'll move on to dose level 3 and 4. And by the time we get to dose level 4, we're actually giving 440 million cells, which is 10 times the amount of cells that the patients that we're going to talk about today received. So this is the data that we saw, and very much from an efficacy perspective, we had very low expectations as this particular group of patients received a very low level of cells, so 44 million cells. We were very pleased to see that 3 patients out of the 4 that were given the cells achieved an overall best response of stable disease. And that's by what we call RANO criteria, and that is criteria specific for neuro-oncology or for brain cancer. What's notable here is that with stable disease, that's actually quite an achievement in brain cancer. Progressive disease means that actually more than 50% of the disease has to actually have gone away to have a partial response, right? And so this means that these patients actually had somewhat less than 50% of the disease disappear, but certainly that's a pretty high bar for patients to meet at this dose level. So very pleased that we saw this 75% disease control rate. And a disease control rate is often used in glioblastoma because we really don't see a lot of complete responses or partial responses. Give you a little bit of a sense of how our disease control rate compares to what else is out there. What you see in this table are 4 different assets, and obviously our chlorotoxin CAR T highlighted in the bottom row in orange. The 3 assets above that are all assets that are currently in development for glioblastoma, and they've actually finished their entire dose escalation. So where we have 4 different dose levels, these assets have actually finished all of their dose levels. You see the disease control rates that they're showing somewhere in the neighborhood of 20% to 48%. And at our very lowest dose level, we're already achieving a disease control rate of 75%. So we are highly encouraged about this data as we move into the higher dose levels, and hopefully what this will mean as we continue to dose patients with more effective doses going forward. This trial was actually set up and designed to really look at safety. And so what we were pleased to see is there were no dose-limiting toxicities, which we actually knew a while ago, so we were able to move on to the second dose level. It was also characterized that generally the therapy was tolerated well by all patients. What we did note was that there was a grade 3 cerebral edema that occurred in 1 patient. Now, what's important to note is that was attributed to what we see as only the possibly level. So when there's a grade 3 event, the clinician actually has to decide, is it probable? Is it possible? Is it absolute that the actual event is occurred by the therapy given? In this particular situation, the clinician assessed that it was only possible that the chlorotoxin CAR Ts actually caused this grade 3 cerebral edema. What we do know, and what the clinician highlighted to us, was that cerebral edema is a very common event in glioblastoma, particularly in patients in recurrent glioblastoma. So certainly this is something we're going to keep our eye on, but it is something that we actually believe may have been disease related more than chlorotoxin CAR T related at this time. From there, we also talked about the bioactivity. And this is important for us, and I'll give you a little sense of why. What we noted, or what was noted in the abstract, was that there was a persistence of chlorotoxin CAR T cells. And what that really means to us is that they were able to see the chlorotoxin CAR T cells days after they were infused in the patient's brain. And that's critical because if the cells weren't able to be detected, it potentially means that they died off. And if they've died off, they obviously can't do their job to find and kill those cancer cells. So this piece of information really tells us that as the cells are entering the brain, they are staying alive, so they're able to focus on finding the glioblastoma cells and killing them. And you hear the conclusion that generally that means that we don't believe that the cells are immunogenic. Immunogenicity is just really when the body's own immune system detects something foreign and then mounts a response against it. So in this case, the body is not mounting that immune response against the chlorotoxin CAR T cells, allowing them to stay alive. So this is really encouraging as well, because we really need those cells to stay alive in the brain so they can do their job. The other abstract, there was a second abstract that was actually released regarding chlorotoxin as well. And this focused on some of the more translational aspects rather than the clinical aspects of chlorotoxin. What we did note in that particular abstract was, again, we talked about the relationship between chlorotoxin binding and MMP2 expression in a patient. And what this abstract really highlights is it's supporting the rationale we have for evaluating MMP2 as a marker for response as we go forward, and also for using MMP2 to determine the best other tumor types for us to go into. And so tumor types that highly express MMP2. So we continue to use this translational data to really look at how do we continue to develop this asset as we move forward, and what are the patient populations that are most likely to respond to chlorotoxin. So some really important information here as well for us. So again, just highlighting the data that came out, early signs of potential efficacy. So that 75% disease control rate, which we are very encouraged by, with the 3 out of 4 patients having a best response of stable disease. A very promising safety profile with no dose-limiting toxicities and generally well-tolerated treatment, and encouraging bioactivity where we saw persistence of the cells and no signs of immunogenicity. So as we continue to move in those higher dose levels, we have lots of reasons for enthusiasm and very encouraged by this initial data set that we've seen. Just giving a sense of where we're going, right? So right now we know that the patients are being treated in that dose level 2 at 88 million cells, and then we'll be moving to dose level 3 and 4 next year at 220 and 440 million cells. So certainly looking forward to that data set. For us, just highlighting a couple other quick things about chlorotoxin before we open to any questions. With chlorotoxin, the phase 1 clinical trial has been a single site trial that has been underway at City of Hope Hospital in California. In August of this year, we were able to get our own IND from the FDA, and what that's allowing us to do is open up additional clinical trial sites next year so that we can accelerate this particular trial. And then looking at moving into the additional tumor types, with our IND, we'll be able to move in advance some of those additional tumor types to clinic as well in 2022. So we'll start to look forward to getting the second phase 1 clinical trial in 2022 in chlorotoxin and moving through the glioblastoma trial. So just to wrap up and highlight a few things before I check in with Matt to see if there were any questions. We're very pleased to be the leader in cell therapy in Australia as we continue to be the only clinical stage cell therapy company. And soon we'll have 3 phase 1 clinical trials in clinic, or phase 1 clinical trials ongoing. Our lead asset continues to advance in the phase 1 clinical trial with what we're now characterizing as very promising early clinical data. The acquisition of CHM2101, so our CDH17 CAR-T that we acquired in July, is making very rapid progress to clinic, and we're planning to open that trial in 2022. And we continue to look into pipeline development, so bringing in additional assets into our pipeline to innovate and diversify our portfolio. And we're doing all this with what we think is the most experienced leadership team by far in cell therapy development, and certainly a group that has the expertise to move these assets forward in development. So Matt, I'll stop there and see if there are any questions about any of the data that I am able to answer. The one thing I might just highlight, Matt, before you let me know if there's any questions, is this abstract, there will be final presentations by the authors at the scientific meeting next week, or I guess later this week now, it's Monday. And if we do bring, or if they do bring forward any additional insights through the context of those presentations, we'll be certainly coming back to let everybody know what that looks like.
Great. Thanks very much for that, Jen. We'll move on to the Q&A. As you've mentioned, there's quite a few have come through. Once again, if you have a question, please submit it using the Q&A function in the Zoom app. But Jen, the first question is, and there's been a couple of questions around this, which is disease control and how long it was exhibited, and whether you can elaborate on that at all.
Yeah, at this time, Matt, I don't have the data on the disease control durability. We are hopeful, Matt, that as part of the final presentations, that the clinicians may be able to address that.
Thanks, Jen. The next question is, can you tell us anything more on how dose level 2 is progressing?
Dose level 2 continues to move forward, Matt, in terms of we anticipate that by early next year, we'll be dosing dose level 3. So we continue to progress and move those patients forward and hope to have that wrapped up shortly.
Great. The next question is, can you expand on the difficulties of recruiting patients in this trial?
Yeah, I think it's a very challenging time to recruit patients in trials, particularly in trials where they have to travel at all. As probably everybody is aware, traveling in this day and age or in the last year has been very challenging. So that's, Matt, probably first and foremost, one of the things that's made it more difficult to recruit patients in this trial is that generally patients traveling can be challenging. And so people are really choosing to look at ways to be treated closer to home. The other thing with this particular trial is the patients come in, they actually do have to look at their MMP2 expression. So the trial does require patients to have MMP2 expression. So we do ask that all patients are screened for that. And then the patients have surgery. And I think for some patients if you think about this disease, most patients with this disease actually only have sort of a 6-month survival expected once they recur. A lot of these patients are progressing very quickly. And so what we're really trying to do is make sure that we're catching those patients before they're progressing to the point that you can't administer cells anymore. So some challenges making sure, Matt, that the patients are eligible and that we get them into the center and then that we get them on the therapy.
Yep, great, thank you. The next question is, when will the 2 new clinical sites or further clinical sites open up for recruitment in the phase 1 trial?
Yeah, right now, Matt, we anticipate that they will be opened in the first half of next year. It's always a little hard to pinpoint exactly when because a lot of it has to do with contracting with the new sites. And sometimes we're held out a little bit at the timetable of the institutions we're working with. So right now, with the way that we're progressing, we do see that happening in the first half of next year, though.
Great. The next question is there any update that we can discuss around how the work with OncoBay is progressing?
Yeah, so OncoBay has been brought in as our clinical research partner, and it is going very well. They are actually working hard with the new sites to make sure that they are all prepared. And so far the work has been very positive and the collaboration has been very positive in getting the assistance that we needed to get those additional sites up and ready.
Great. The next question is, by no dose limiting toxicities, do you mean there are no indications that there will likely be any problem proceeding through the dose escalations?
No, so the term dose limiting toxicity essentially means that in dose escalation, there's the 4 different dose levels that we give, that we're giving. In each of those dose levels, what we have to do is after the last patient is dosed in that dose level, we have to wait 28 days. And we then have to assess whether any of the patients had adverse events that were so serious in nature that they would be considered what we refer to as a dose limiting toxicity. If there were very serious in nature adverse events, essentially that would stop the trial or make us go back and dose patients in that dose level again. So when we're talking about a patient, this group of patients having no dose limiting toxicities, what we're saying is in those 4 patients, there was no serious events that actually warranted us stopping the trial or needing to go back and look at that particular dose any further. We're able to continue to move on to the next dose.
Thanks, Jen. The next question is, what proportion of recurrent tumours are MMP2 plus?
Yeah, it's a great question. And one of the reasons that the work that Dr. Barish is doing, that translational work, is so important. He is currently looking at the data they have. What we do know is MMP2 expression generally is probably somewhere between 70 and 80% in glioblastoma patients. Matt, what Dr. Barish is working on now, and hopefully we'll have sometime next year, is whether or not we see any difference in that percentage in the recurrent patients in this trial.
Thanks, Jen. The next question is, how relevant is the location of the tumours in these patients? Is the mode of drug administration a limiting factor depending on where the tumours are located within the brain?
Yeah, it's very important. And I think particularly at the early dose level, where we were only giving patients cells intratumorally, right? And we know that glioblastoma doesn't tend to limit itself. So I think as we move to the second dose level and continue to the third and fourth dose level, where we're giving cells both intratumorally and interventricularly, we believe we'll have better coverage, so across the brain. So from that perspective, I think that is where we think that we'll have a better shot or a better chance of being able to affect more of the glioblastoma.
Thank you, Jen. Another question that's come through is, how many patients need to be enrolled in the trial overall for the data harvested to be deemed statistically significant?
Yeah, so this is a single arm phase 1. So we're really not comparing to anything, right? So we don't have, it's not like a phase 3 trial where you have a comparator arm and that you're looking for a difference between the two arms. What we're really looking for here is to be able to assess, is it safe? What is the maximum dose that we can give patients safely? And what type of efficacy comes with that? So there's really no sort of p-value or statistical significant value, hazard ratio, or anything we're looking for in this particular data set because there's no comparator, Matt. I think what we do look at importantly, obviously, like I showed you, are some of the analogs to how other assets do in the same type of patient population. And then as we look at those higher dose levels, we'll really start looking as well at what the progression-free survival is of these patients and potentially what the overall survival is as well in these patients. Because we know in the recurrent setting, progression-free survival, the average is about 10 weeks. So it is very low progression-free survival we see in these patients. And the overall survival is only about 6 months. So we'll start to look at those efficacy endpoints in more detail as we increase to the higher dose levels, Matt.
Thanks, Jen. The next question is, do you have to wait for recurrence following the surgery before administering the drug, or at what stage is the drug administered?
Yeah, so what happens with patients is they're diagnosed, they receive a first set of therapies, whatever that be with their physicians. It's generally some form of surgery, radiation, and temozolomide. And then basically what we see is, around 90+% of patients recur. So once a patient recurs, that's when we would start to look at them as eligible for our trial. That's when we would assess and take a look at their MMP2 expression to confirm that they have MMP2 expression. And then generally what happens is that there is a second surgery, and that's when the catheter is placed as well. And then after that is when the chlorotoxin cells are administered.
Thanks, Jen. A question that's come through is with regards to the other trials that you mentioned and whether we know what sort of disease control they showed at early dose levels.
Yeah, and you know what? We tried very hard to be able to find that data, Matt. We were not able to find just dose level 1 data on those analogs. But I think if you consider that, 20 to 50% disease control, including your higher dose levels, I would assume at the low dose levels, it was either not existent or very, very low, Matt.
Thanks, Jen. We've got time for just one more. There's been a lot of questions, so thanks to everyone for sending those through, and we'll come back to anyone that we've missed via email or separately. But Jen, one final question was, do we have a plan on how we'll be updating in terms of the level 2 data? Will it be another conference presentation or something separate?
Yeah, Matt, I think at this point we have not confirmed the plan. I would anticipate that we will be able to confirm that sometime in the new year. The authors, obviously, Dr. Beatty, who leads this trial, and Christine Brown, are very excited about being able to share this data with the scientific community. And so the next major scientific meeting is ASCO, which is always the first week of June every year. But we'll obviously be in further discussions with them about the right timing for all of the next sets of data release.
Great. Well, thanks for taking all those questions, Jen. I'll just throw it back to you for a closing comment before we finish up.
Yeah, thanks, Matt. And thank you for all of those rapid fire questions, everybody. I greatly appreciate the interest and the enthusiasm with all of the questions. As Matt said, anything that we weren't able to get to, we certainly will try to follow up by email, but please don't hesitate to contact us directly if there's anything else. You know, I think maybe just the last words on this is we certainly are very enthusiastic and very encouraged by what we saw in this early data set. We truly believe that the asset is showing some very promising activity and you know, a promising adverse event profile to go with that. So we'll look forward to the final presentation of the data at the end of this week at the SNO Congress and see what type of questions or if we learn anything additional as that data is presented by Dr. Brown and Dr. Barish. And we'll certainly provide any updates on anything additional or any additional insights we have from that data set as we go forward. So I thank everybody for taking the time this morning to come and have me take you through the data. I truly appreciate that. And certainly we appreciate all of your support of Chimeric and of chlorotoxin and look forward to speaking again soon.
Thanks very much, Jen, and thanks to everyone for joining today's Chimeric Therapeutics Investor Webinar. You can now disconnect.